Okay, well, thank you. I want to welcome everybody back to continue our exciting round of talks. As I mentioned before, we've got Tom Kash joining us. We're thrilled to have Tom with us. Tom is at the University of North Carolina at Chapel Hill and in the Department of Pharmacology doing exciting work related to another component of the circuitry that we'll hear about, which is the extended amygdala. He'll tell you all about that. His work has been focused on substance use disorders, but also anxiety. And what I find particularly interesting is the interface between aversive and appetitive and reward related behaviors, which is so crucial when we think about all different types of psychopathology in relation to appetitive and aversive drives and reward related circuitry. Tom is also the vice chair for faculty development in his department, and as like Guo Ping, has won many awards, which I won't mention, but you can see when you read his bio. Tom, thanks so much for being with us. It's wonderful to have you here. Take it away.
Well, thank you so much, everybody, or Ned and Richie for inviting me. It's when I look at the speaker list, I feel like I'm, it's that first Thanksgiving where I'm allowed to sit at the adults table, because the other speakers are just amazing, doing such fantastic work. So what I'm going to talk about today relates exactly to what Ned brought up, is really the idea of trying to understand some of the interactions between substance use and aversive states. And in particular, I'm going to focus in on a modulator, serotonin, or I'll call it serotonin or 5-HT. And so I think something that is really on the forefront here is data from a study by the Brookings Institute. It's several years old, but I think what it highlights is that, if we look simply from the year 2000 to 2014, there's been a dramatic increase in the death rate by alcohol, drugs, or suicide. And, they refer to this as the deaths of the despair. And I think, what, in my mind, what this highlights and also really hits home is that, before the COVID public health pandemic crisis, there actually was a mental health crisis as well relating to psychiatric disorders and substance abuse, alcohol and opiate use disorders. So I think this is something that is critically important for us to understand. And where I really, my long-term interest in understanding this and studying this is the idea that substance use, in particular, I'll talk about alcohol consumption today, has this interesting bidirectional relationship with aversive states. One being that, in the presence of aversive states such as, heightened anxiety or hyperarousal or chronic pain or, general dysphoria, alcohol has been used to relieve these symptoms. It can kind of normalize how one feels. And so it's the idea of self-medication. The downside of this is that with excessive alcohol consumption, you can actually get a potentiation of these aversive states. So excessive alcohol use can drive changes in anxiety, pain, hyperarousal, and other things. So really what I am interested in doing in my group is using animal models to parse out the interactions between these, try to identify mechanisms by which we can intervene in this process. And if we get down to the details, what I'm interested in is modulation. And so just a quick primer on modulation. Let's imagine we're looking at a cell. And so this is a neuronal cell in the brain. And for the sake of argument, I'm going to ignore astrocytes, glia, everything that's non-neuronal. This doesn't mean it's not important. It means I don't have the bandwidth to talk about it and give it justice. But we have this idealized cell, and this cell receives a inhibitory or GABAergic input, and then excitatory or glutamatergic input. On some level, the balance of excitation and inhibition will govern the output of this cell, the timing between these two as well. But there's another layer of function in the brain, and that's looking at modulators such as neuropeptides. Corticotrophin releasing factor, CRF, is one that I've studied in my lab for a while and before, but also biogenic amines like serotonin, for example. And these modulators work via G protein coupled receptors. And these G protein coupled receptors activate signaling cascades, and they can be located at various points in the circuit. They can be on the cell we're studying to either excite or inhibit. They can be on presynaptic inputs where they can increase or dampen release dynamics. And so they're really critically positioned to, the way I think of it, they're kind of analog control agents for specific circuits in the brain. And, in particular, GPCRs or G protein coupled receptors are important because they represent a means by which we can pharmacologically intervene. So we can develop treatments that target these systems if they're dysfunctional, or we can develop treatments that will act in opposition to these dysfunctional systems. And so what I am going to focus on today is serotonin. And so this is, one of my favorite systems to study. In part, serotonin is the system that people say can do everything, but it's too complicated. There's many receptors. And I think I embrace that complexity, but also work to narrow it down. So when we think about the areas we're studying, serotonin has come up over the years. In particular, work from John Crystal's group showed that MCPP, this is a agonist that acts on 5-HT2C receptors, among other things, can actually induce craving in alcoholics. Further, so it kind of suggests that activation of 5-HT2 systems plays some sort of role. In addition, work from Hank Kranzler's group showed that SSRI treatment, so serotonin selective reuptake inhibitors, can increase anxiety and alcohol consumption in a subset of alcoholics. So kind of lending some credence to the idea that increasing serotonin can drive sort of a negative outcomes. I would say that in some people there was the opposite effect. So this is an opportunity for perhaps understanding individual variants as well. And so from the more generic perspective, there's abundant literature out there on how in the early phases of SSRI treatment, that can lead to anxiety, panic, and suicidal ideation in people. And then if we study it in rodents, we can also identify aversive behavior. And there's a really great review by Bergert and Bauer on this from several years ago. And so if we take a step back, so the serotonin system is complex. There was a really elegant paper by Jing Ren, who was a postdoc and now has her own lab at Oxford, I believe, or Cambridge. Now I'm blanking. But what she did was identify these subsystems of serotonin neurons and really focused on these two major subsystems. We had these frontal cortical projecting serotonin neurons that were activated by reward, inhibited by punishment, and her behavior suggested they promoted active coping behavior. And then we had these amygdala projecting serotonin neurons, which were activated by both reward and punishment and promoted anxiety-like behaviors. And the way we conceptualize this is we have this, somewhat of a positive serotonin system and a negative serotonin system. And that's we're painting a very broad picture there, but it's helpful for narrowing down and conceptualizing. And so we're actually looking at both of these systems. So Melanie Pina is a postdoc in the lab who has a K99 who's on the job market for anyone interested. And she has some really exciting studies looking at these frontal cortical projecting systems and how they regulate alcohol use disorders and related behaviors. And then Meg Flanagan, another postdoc in the lab, has been focusing on these amygdala projecting or these aversive serotonin networks. And so today I'm going to talk about Meg's work focusing on this amygdala projecting system. And so with serotonin, this doesn't come from a vacuum. There has been data suggesting that serotonin signaling in this aversive subsystem is implicated in particular in alcohol abuse. And so some really recent data suggested 5-HT2C receptor signaling. So we're narrowing down from 5-HT2 receptors to the 5-HT2C receptors. That 2C in the lateral habenula, a structure that is really interesting for aversive behavior, was linked to withdrawal-induced anxiety in rats. And so we have this receptor in this region that seemed to be important. And then work from my own lab by postdoc Catherine Marcinkiewicz, who is now faculty at University of Iowa, showed or had data suggesting that 2C signaling in the bed nucleus of the stria terminalis. I call it the BNST. I know other folks call it the BST, but I'm giving a talk, so it's BNST. And 5-HT2C has been implicated in withdrawal-induced disruption of social behavior, and this is using a dependence model. Alcohol dependence model, rather. And then more generically, some work from Catherine and Chris Mazzone, who was a postdoc, who was a grad student, now a postdoc with at NIEHS, showed we did a bunch of, I would say, mechanistic circuit work looking at how serotonin inputs in the BNST alter behavior and function. And we found that we can get enhanced fear recall, enhanced avoidance behavior related to the activation of the 5-HT2C receptor. So it kind of puts on the forefront these two regions and the 5-HT2C receptor. And so that is really what we're focusing on. And just stepping back to the circuitry of interest. And so the BNST is where I've spent most of my independent scientific career for the past dozen years, and then before when I was a postdoc. And this is, the Kalin group has done an amazing job probing the BNST in the non-human primate model. And really, I think there's just a huge amount of work suggesting that the BNST is this critical anatomical substrate for interaction between aversive states and reward. It has all these really interesting bidirectional connections with hypothalamic, midbrain, and hindbrain structures. It receives cortical and thalamic and input from other amygdala regions. So it's this really critical hub for regulation of kind of avoidance and reward. The habenula is also a really interesting structure, a little bit newer for us, so it doesn't get center play in the figure. It has numerous connections throughout the brain. I think the two that come when I think of the habenula, there's this really robust connection to the ventral tegmental area, where it's a negative regulator of dopamine neuron function. And then there's also an output to the dorsal raphe. And critically, the dorsal raphe, this is the source of serotonin for much of the brain, projects to both the lateral habenula and the BNST. So we're kind of setting ourselves up to understand how these two circuit nodes connected to aversion and reward might be playing a role here. And so briefly, I'm going to talk a little bit about the model that we're using, and then dive into how we're probing the role of serotonin and serotonin signaling in this model to study this. Okay, and so the model that we're using is called Drinking in the Dark. It is a relatively simple model. It leverages the idea that mice, so we're using all mice, they will drink more in the dark. And so what we do is we give them 2 bottles in the dark cycle of water and a 20% alcohol solution. And we do this, it's a 4-day procedure. So on Monday through Wednesday, they get 2 hours of access, and on Thursday, they get 4 hours access. And so we do this. So what Meg did here is repeat this for 3 weeks. And then we did a 1-week abstinence period, and then looked at a variety of behaviors. And we focused on social behaviors, open field just for a generic measurement of behavior. And then we looked at acoustic startle, something we thought was really interesting because it is a translational assay. You can look at startle response in people. And also it can provide a lot of insight as to the nature of dysfunction, independent of locomotive confounds for many other tests that we will conduct. And so the data looks like, so we did all of these studies in male and female rodents, mice. And this just shows, so here, the females are in the bluish color, the males are in this green color. And really just to highlight, indeed, mice will drink relatively large amounts of alcohol over this procedure. They hit a blood alcohol concentration above 80 mg per deciliter, and so that's equivalent to 0.08 in people blood alcohol level. And so this is a really high level of intake. And an important thing to make note as well is when we look at the intake normalized by body weight, females always consume more alcohol. And so this is essentially, when we give free access, this is a variable we have to consider when we're thinking about these studies. And another variable that we need to consider with these studies is all of these animals are single housed throughout the entirety of this experiment. And so, I think we're all a little bit more conscious of social isolation as a variable given the past year. But I think that's something that we need to consider with these results and how that might interplay or impact some of our findings. And so mice consume alcohol. So the first question was, does this impact sociability? And so previously we found in a alcohol dependence model, which is a much more severe high level forced exposure model, we saw deficits in social interaction. With this model, what we saw was actually, if you look at the first graph, this is social preference. So we're using the 3-chamber sociability task. And so the first part of that is just looking at the preference for the mouse to interact with another mouse versus just an object. And you see there's no difference here. All of the mice, both male and female, water controls or drinking mice, alcohol drinking mice, all show a social preference. There's no effect of anything here. Social novelty, you get the mice a choice between a mouse they already met and a novel mouse. And you can see here in males, there's no difference here in the treatment group. However, in females, we see that there's a reduction in this social novelty preference. And so there is this, it's a clear effect that's significant that we found is repeatable over time. But it is somewhat subtle. It suggests too, it's not just a pure difference in social behavior, but there's something more here. There might be some social identification differences that we're observing. So there's this subtle social defect, which is only in female mice. Then we looked at open field behavior. And so the idea with the open field assay is that you take a chamber, moderately bright light, and then you let the mice explore, and they have an innate aversion to open spaces. And so if they avoid open spaces more, so if they avoid the center more, we might conceptualize that as an increase in avoidance-like behavior or increased anxiety. And we do see that for males. A trend in females, if we look at entries to the center, it's a alcohol effect in both groups. Also, we see some differences in how much they move and their mean velocity. So there's a little bit of a locomotive deficit here. And that's not inconsistent with some previous reports of alcohol withdrawal where mice just seem to move slower over time or move less over time. I'll add that this particular effect has not been as robust across multiple cohorts of mice that we have done, as you'll see in the additional experiments, but it was present here. And finally, we looked at acoustic startle, and we were excited by this. We're excited to use this because it's a repeatable assay, and so many of the assays for we're supposed to call it avoidance-like behavior are not as readily repeatable. Whereas startle, we can repeat it over time, and so it gives us the ability to track behavior. And also, we know in people that suffer from PTSD, we can have increased startle responses, and folks who suffer from depression, we can have reduced startle responses. So it's a relatively good sort of way to parse out endophenotypes or even just behavioral phenotypes. And so we looked here. What we found in males, there was a significant increase in the startle response at the highest sound level. In female mice, there was no effect. And so this was 1 week out. We wanted to see if this was a persistent change. And so what we did was what Meg did was look at 28 days, and we saw, I was pretty excited to see that this effect persisted in the male mice and still no effect in the female mice. And so then, if you have something that lasts 7 days, that lasts 28 days, then I don't know, we thought it would be interesting to see what happens when we go out 8 weeks. And so we again looked at 8 weeks. So 8 weeks into abstinence, we see that there's this persistent increase in startle response in the male mice. And I'm not going to show it, but there still was no impact or no difference in the female mice. So we have this behavioral paradigm where we can, it hits certain buttons. We can see sex-dependent differences of differential impact of alcohol consumption on behavior, so sex differences in behavioral outputs, but also we can assess alcohol intake in this model. And that's helpful where we can start to attempt to link some of these behavioral differences and plasticity events to the alcohol consumption itself. And so that is, this is the idea that this model does give us a bit of capacity to look at this bidirectional relationship with some ability to do mechanistic manipulations and assess dynamics of circuit elements. And so we have this DID model, and the next question is, what do we do with it? And so now we shift our focus to looking at serotonin and assessing serotonin function. And so quickly, I thought it would be good just to highlight some of the tools we have in hand to probe serotonin systems. and then march through some of the data that we have collected. I'd say this is all unpublished data that we're really excited about, and hopefully we'll be finishing up this project or a first manuscript soon. And so for the tools we have, we've been interested in serotonin for years. This was my first R1 was on serotonin, the BNST, and anxiety, and so we've kind of kept up this stream of science for a while now and developed and obtained a number of tools. So we have mice 5-HT2C receptor Cre mice, and so these are mice that have a Cre recombinase in the 5-HT2C receptor locus, and that allows us to genetically manipulate using chemogenetics or optogenetics and monitor using fluorescent, for example, GCAMP, which is a genetically encoded calcium sensor. So we can manipulate and monitor this population of neurons. We also have a floxed 5-HT2C receptor mouse, and so this allows us to virally delete this specific receptor in discrete brain regions. And so we have the ability to ask what happens when we delete it from the BNST or from the habenula. We also have been working with a number of groups to to test sensors for serotonin that they have developed. So we were part of a great collaboration with Lynn Tin's group recently, looking at a sensor, and we have another sensor, GRAB 5-HT receptor. So this is a receptor from Yulong, or a sensor from Yulong Li's group that is based on the 5-HT2C receptor, and so we can use this to measure the dynamics of serotonin in vivo, and we have some fresh, hot off the press data showing some interesting developments from there. And then we also have a couple mouse lines for genetically targeting serotonin neurons. And so we have this toolbox that let us really dig in and understand this system. And one of the first questions we had was, Jing Ren's data suggested the presence of this aversive or amygdala projecting subsystem of serotonin neurons. So we wanted to, just confirm or ask the question, what is this subsystem conserved between specifically the BNST projecting dorsal raphe neurons and the habenula projecting dorsal raphe neurons? And so what we did was some anatomy where we injected a retrograde tracer, a fluorescent retrograde tracer, into both the lateral habenula and the bed nucleus of the stria terminalis. We focused on the ventral BNST for this because that's where the densest serotonin innervation is. Gave the tracer some time to go back up to the raphe, killed the mice, and then stained for serotonin in the dorsal raphe, and then just looked at the overlap of these two retrograde fluorescently labeled indicators and the 5-HT in the raphe. And what it told us is, in the graph shown in green here, that most of the neurons from the dorsal raphe to the habenula and from the dorsal raphe to the BNST are indeed serotonergic. So there's a strong serotonin projection to these structures. And then if we look at overlap, if you look at the neurons that project to the habenula, we see that most of them also project to the BNST. So there's the idea of this, there's a similar sort of routing of information, parallel routing of information between these structures. And when we look at the serotonin neurons that project to the BNST, we see that there's a little bit of a difference here. Maybe about half the cells also project to the habenula. So there might be differential routing. Also, it might be that the BNST is actually just bigger and we didn't quite, it's really hard to fill up both the ventral and the dorsal with tracer accurately. So we might just be a sub-targeting of the ventral domains. But we have these, I think the idea that's helpful here is we have these converging parallel sources of information or of modulation to these structures. And when we look at the neurons themselves, so we have, this is in situ looking at the 5-HT2C receptor and then markers of GABA neurons. So VGAT's a marker for GABA neurons. VGLUT2 is a marker for glutamatergic neurons. And we can see in the BNST, it's mostly GABA neurons overall. And then when we look at 2C, it's mostly overlapping in GABA neurons. So it's primarily on GABA neurons, whereas in the habenula, it's the opposite. It's primarily overlapping with glutamatergic neurons. And so there's an interesting, it kind of sets up this interesting question. We have this parallel modulatory influence regulating two different types of cells. And, how is that, what is that doing? How do they project out of those structures to regulate behavior in a meaningful way? And we have thoughts on that. But it does set up kind of an interesting conceptual question. And so having said that, the first sort of question we wanted to get at was how do these neurons actually respond in these structures during alcohol consumption, during social behavior, and during startle. So focusing on these three behaviors that we think are important and appear to be modulated themselves by alcohol consumption. And so the approach we took here was a fiber photometry approach. And so with this, we took our 5-HT2C Cree mouse line. We injected a Cree-dependent GCAMP 7F. And so what this is a viral approach that will infect neurons in a Cree-dependent fashion with GCAMP, which will, when the cells, the idea is when cells fire, you get an influx of calcium, and we get an increase in fluorescence as calcium goes up, and thus measuring changes in fluorescence are a proxy for the activity of these neurons. It's not perfect, we know that, but it's it's a good way to kind of look at the big picture. And we're looking at the population of neurons here. We're not able to digest down to individual cells. And what we did was ran the mice again through the drinking in the dark paradigm, a week of abstinence, and then conducted a series of behaviors. And so what we looked at here, so the first assay we did was just a free social interaction. So we parsed this out, or I'm going to parse this out by female mice and then male mice, because there are some interesting differences in behavior and also dynamics that you'll see as we do this. And so when we looked at just this first social interaction between mice, you can see, and so we're only measuring responses in the mice that was exposed to alcohol. The other mouse is just there as kind of a novel character to interact with, novel mouse to interact with. And we can see that the first thing you can notice is that activity goes up in these neurons when there is this social interaction. And you'll also notice that when we look at the water versus the did group, there's no, as of how we're analyzing it now, there's no significant differences between these groups. The shape of the response after alcohol exposure looks a little different, and this is something we're digging very deeply into using additional refined methods to get at. But with this first pass analysis, no large differences in the habenula. When we look in the BNST, we see again there's this positive increase, so an increase in activity cells, this population of cells, but no difference, no alcohol impact on that behavior. So these neurons appear to be recruited by this, or activated in response to this social event or social mouse, but no differential effect. And so then we looked at alcohol consumption. And so in the habenula, what we actually saw was that, so in water mice, when they were first exposed to alcohol exposure, there really was a negligible effect here. So not really much happened. However, in contrast, with a history of alcohol drinking, we see that there is a robust reduction in the response to alcohol. And so this was actually really fascinating for us, because it highlights this idea. So this population, the habenula is often thought to be an aversive structure, and activity there can drive ongoing aversion. Really the idea of alcohol negatively regulating that in a alcohol drinking history dependent fashion suggests that this might be a key regulator of negative reinforcement. So this is something we're really excited about and digging deeply into. However, in the BNST, we saw that there was no real difference here. A little bit more heterogeneity of the responses per animal, but net reduction in activity between both groups. So in the BNST, alcohol drinking appeared to negatively regulate the activity response in these cells. And so, one of the things that we did to get the mice to drink a little bit faster was we did a mild water deprivation. And so part of what the question one might ask is, are these responses just related to the mice being really thirsty and then having a chance to drink water? And that's, I think, a really valid question. And so what Meg did was just look at responses to water, and then found, I think really interestingly, that water itself was able to actually cause a reduction in this response in the habenula in both groups. So, if you're thirsty and you have water, it was sufficient to reduce this sort of thirst-driven aversive state, which I'm overreaching with that, but I think it's a good good discussion point. In the BNST, we again saw a little bit of, or a fair amount of heterogeneity, but no difference between the groups. And I'll say in the BNST, I think part of what we're seeing might be due to precise placement of these fibers. So we're working through that and characterizing it a bit better as we go through. But it highlights this response to alcohol really may have been a negative reinforcement signal. And so we looked a little closer at the relationship here. And in the habenula, we saw that this the signal upon drinking, this negative signal, was actually strongly correlated with both the cumulative alcohol intake and the average daily intake. And so that kind of raises this idea that maybe this is, in female mice, maybe this is an interesting sort of negative reinforcement signal. In contrast, the BNST correlations were not significant and really not that informative overall. So, we're seeing a difference in how information is being dynamically routed through these circuits or these populations of cells. Finally, in the female mice, we want to look at startle. And so again, as we saw before, there was no significant change in startle response in female mice after the drinking paradigm. And when we looked at the dynamics, again, we see that as opposed to the alcohol drinking or water drinking, which showed a negative deflection, we see a positive deflection, so an increase in activity here in both the BNST and the habenula, but no effect of DID on these responses. So this was a conserved response, but invariant and not plastic in looking at these measurements. So then what we did, we wanted to switch to the male mice, and again, same exact paradigm. I'll go slightly faster because it's a similar flow of information. So we looked at novel social targets in the habenula and BNST, and again, we see a similar pattern where there's this increase in activity in these neurons in both regions that is not modulated by alcohol exposure. And in the kind of nifty thing, and this is fresh off the press data, we've been working with some of these new sensors, and so we only have it in male mice, but we looked at 5-HT release using the GRAB 5-HT 3.5, and this, when serotonin is present, you get fluorescence in these with this sensor. And so in the habenula, we saw evidence for an increase, so social interaction induced increase in serotonin in the habenula, as well as the BNST. So this was kind of exciting. These are just naive mice, and so we're hoping this gives us an opportunity to look at the in vivo dynamics of serotonin on the level of serotonin release. We'll see how well that actually works, but I thought this was worth including because it's pretty exciting to us. But back to the kind of normal, or back to what we saw before, looking at responses to alcohol. No difference. As opposed to the female mice, there was no evidence for any plasticity in the male mice in the habenula or the BNST. We get this negative deflection in both circumstances. When we look at startle, we see, as before, in this smaller group, there's this increase in startle response. And when we look at the response, kind of interestingly, we see an increase in the size of the startle response after alcohol exposure in both the habenula and the BNST. So it highlights, to me, it's kind of an interesting sort of difference, and we're still working to understand why, but alcohol exposure that does engender these sex-dependent differences in behavior is having some sort of neural activity-dependent changes as well. So we're thinking that there is some sort of difference in plasticity or activity that's happening here, rather. And so just to summarize, social interaction is driving this increase in activity, invariant from all groups. In the females, we see this alcohol suppression only after a history of drinking, which we think might be evidence for a negative reinforcement. And then we don't see that in male mice or the BNST. And then we see this potentiation of startle response and startle neural signature in males in both regions. So highlighting, this, in a sense, this really gives us the idea that the behavioral differences that we're seeing in male and female mice, they track on to in vivo difference. And this is interesting and important because when we look, collectively at the larger data set involving men and women and the comorbidities and reasons why they might drink, there are differences there. It's not a heterogeneous sort of response. Everyone drinks for this region or that region. You start to parse out some critical sex differences in responses. And so with the dynamics, kind of having a handle on what's happening, we then wanted to look at, look a little bit more. So when we look at these neurons, it doesn't really tell us what the receptor is doing. And so we wanted to ask, what is this receptor actually doing here? And so what Meg did was delete this receptor. And so she's been working to delete it both in the BNST and habenula. Right now, we only have the BNST data set, so that's what I will go through. There's a lot of interesting tidbits here. And so this is the Floxed 5-HT2C mouse. We got this from Joel Elmquist at UT Southwestern, and you can see that when we inject a Cree virus into the BNST, we can indeed get a reduction in 5-HT2C mRNA. So the Cree Floxed mouse system works well, as many others have shown. And so what Meg did was took mice, either injected Cree bilaterally or just a control virus bilaterally, waited 4 weeks, and then did the same experiments that we did before, look at drinking, look at sociability, look at startle, and look at open field behavior. And this is just a picture showing the targeting. We're really focusing on targeting the ventral BNST versus dorsal BNST here. And so again, parsing it out by female mice first, and then male mice. What we saw, I think somewhat surprisingly, was in the female mice, when we deleted the 5-HT2C receptor, we saw an increase in alcohol consumption compared to the GFP groups. And you can see with cumulative intake and also average intake. And so this was, one of the things when you study alcohol does have a appetitive component. And so it's always important to parse out, is this difference you're seeing simply due to a difference in reward-seeking behavior or appetite? And so we looked at sucrose intake here. And what we found, there was no impact on sucrose intake in this group or of this manipulation. So it wasn't just an appetitive change that we saw. So this was consistent with the idea that there's some sort of, selective effect, at least when we look at sucrose versus alcohol, of this manipulation in female mice in the BNST. And so then looking at social preference and social novelty preference. With social preference, we saw this interesting phenotype that is close but not quite significant, p-value 0.08 for a main effective virus, where there's a general reduction in social preference. And when we look at social novelty, it gets a little bit more complex. Again, we're seeing evidence for this reduction in social novelty preference. But when we delete this receptor from water mice or our control mice, we actually see a significant reduction in social novelty. And then when it's deleted from the drinking mice, we see a restoration of this deficit. So it's this kind of complex relationship, and there's no real simple way to explain this. I think the simplest way is the idea of, it's kind of, there's an inverted U where there's a sweet spot for how serotonin can influence behavior. And if there's too much or too little serotonergic activation in this structure, you can get aberrant behavior. And what we're seeing is kind of these various conditions, we're sliding the scale of where serotonin may reside, and kind of the Goldilocks principle of you need just the right amount to kind of promote the naturalistic or wild type behavior. Again, we wanted to look at startle just to be, kind of follow up through everything. In female mice, I'll remind you, we didn't see any impact of drinking in female mice before. We still don't see anything in female mice when we look at the DITA impact, and we also see no impact of deletion of this receptor on startle response in female mice. So consistent, we have this consistent sort of idea where, this increase in startle, maybe a hyperarousal phenotype, appears to not happen in female mice and doesn't, you can't really block something that's not happening. So the manipulation has no effect. And so this is what this data looks like in a quick summary. And so then repeated the same thing in male mice. What we see, and so we have some more mice here. We haven't tabulated all the data, but the results are consistent, where we see no impact on alcohol consumption in male mice. So again, we're seeing this difference between males and females in the male and female mice in the BNST, where deletion in females causes an increase, where deletion in male has no impact on this consumption. And then also we looked at sucrose intake and saw no impact here either. When we looked at social preference in male mice, we didn't see anything with DID itself. And then when we look at these manipulations, nothing happens on top of that. And when we look at the social novelty preference, again, we see no strong or statistically significant phenotypes that pull out of this. So again, there appears to be this differential effect on social behavior and perhaps motivational alcohol-related behaviors between males and females. And finally, with startle, in the male mice, what we found is we recapitulated the phenotype of drinking on the startle response, and that's shown in the gray-filled circle where the DID EGFP group compared to the control group. And interestingly, when we deleted this receptor, we see that we're able to normalize this change in the alcohol drinking group, and we actually cause a reduction or a near significant reduction in the control group as well. So it suggests that in male mice, at least, 5-HT2C signaling plays a role, absolutely is playing a role in this alcohol-induced hyperarousal response, whereas at the baseline state, it may be playing a role. I think there's a little bit of variance there in the levels of responses. So it's a strong trend though. And so when we put this data together, we really get this, it's kind of a remarkably significant or selective impact in the male mice. Essentially, nothing really happens different except this change in the startle response. And so it highlights that, a 2C might just be aberrantly engaged or engaged in males only for the startle response. So it's really a sex-dependent engagement process, but also it tells us there's some unique plasticity or dynamics or ongoing information routing different between males and females here. And this is something we're really interested in understanding. And so we also did a pretty extensive chemogenetic manipulation of 5-HT2C neurons in the BNST, but because of time and the habenula, but because of time limits, I'm not going to get into this. I'm happy to talk about it in the Q&A if anybody wants to, but there are some kind of interesting consistencies and some interesting differences there that arise. But that's also ongoing work. And I think this really brings us to the idea that, where we are, what's the data telling us? In female, we see idea that there's some sort of engagement in the system that might be altered by alcohol consumption. In female, or in the habenula in female mice, we really see this idea for a negative reinforcement. And this is something we're really excited at our parsing out using the genetic approaches more clearly. Whereas in the BNST, we see this really clear idea that there's a startle response difference and it's gated via serotonin signaling here, or 5-HT2C signaling. Whereas in the female mice, there appears to be more of a motivational component with alcohol drinking and social behavior. And so we have many ongoing studies looking at all these things, as well as doing additional sort of mechanistic studies using slice physiology, as well as downstream, looking at how downstream signaling partners might drive these behaviors. And this is just another sort of model for how we think this might work, whereas the RAF-A appears to have some differences in engagement between male and female mice. And this is consistent with the idea that there are sex differences in how the dorsal RAF-A and serotonergic neurons can work. And with that, I will close it out and say thank you to all the funders, primarily NIAAA, who has been a long-time funder of this work, Meg Flanagan, who, as the postdoc who's worked on this, the many collaborators and other folks from the lab who have helped push this forward. Thanks, everybody, for your time.
Tom, thank you very, very much. Another great talk, and really cool to see these sex differences, which obviously have potentially important clinical, translational opportunities from the standpoint of thinking about the differences in expression and also maybe even in treatment. So, Richie, do you want to lead off with our discussion?
Sure. Yeah. Thank you, Tom. It was really a great talk and great information that you covered in a short time. Nagal Agarwal, who is one of our trainees, is going to lead this discussion, and he will ask a set of questions that have been aggregated among a number of the trainees.
Kash, thank you so much for the engaging talk. So we have a couple of questions that cut across some of your previous work and also the work you've presented today. So our first one was your work, including the recent paper by Lara Hua in 2020, examines the effects of long-term drinking followed by prolonged abstinence. There's also previous research that's shown stress responding among individuals with a history of alcohol use can differ based on whether the individuals are currently in a state of deprivation versus early abstinence. How do you think your data can parse whether the altered stress responding seen in some of your data is due to long-term alcohol use or withdrawal / abstinence?
Yeah, I think it's a good question. I think that it can be really difficult to dissociate. So partially we have to be really specific. What do we mean by withdrawal, right? And so there is acute withdrawal that individuals go through. And, you know, obviously acute, I'm not a physician, but acute alcohol withdrawal can kill you. It can be deadly. It's something that can be medically managed, and it should be medically managed. So there's kind of the acute withdrawal that we see. And then, you know, how does that, you know, I think the verbiage for transitioning what protracted withdrawal versus abstinence might be is important. So I don't think there's really, when you have any model that has, you know, you have this high-low sort of cycling of intake, you're going to have cycles of exposure and withdrawal. So you can dissociate acute withdrawal from protracted withdrawal, or from protracted abstinence, but you can't dissociate the fact that was it withdrawal that actually caused these long-lasting changes. And so there's actually a set of really elegant studies from George Breeze, who is now retired, but he was a colleague at UNC Chapel Hill. And, you know, his model is basically that repeated withdrawal drives this kindling response, that repeated bouts of withdrawal are causing these long-term changes in behavior. And he actually had really nice data suggesting that 5-HT2C signaling played a role in this process. And so, you know, what I think is the long-lasting changes that we see in individuals who suffer from alcohol use disorders, as well as our animal models, are actually, they're because of these repeated bouts of withdrawal. And so it's not, there's no easy way to parse out other than saying, I think that they play a role. And I don't know if that answered the question really as well as I could have, but we can work from there as a starting point if you want.
Yeah, no, thank you. That's helpful.
Okay.
A somewhat related question. So your data also demonstrate, like you just talked about, the role of the BNST in CNS stress responding, but there's also that there's positive and negative feedback loops among central and PNS stress systems, like the autonomic nervous system and the HPA axis, especially in individuals with histories of chronic alcohol or drug use. So how might serotonin receptor systems and also the kappa opioid receptor system, which you've explored in your work, be indirectly affecting stress responses via actions on the sympathetic, parasympathetic, or HPA axis stress circuitries? Yeah.
No, it's a great question. You know, at one point we started dabbling in some autonomic nervous system stuff. It's harder in mice than we thought. So we don't have any really great mechanistic answers there. I will say that Sophia Niera is a grad student in the lab, and we do have an ongoing project where we're looking at PVN CRH neurons in some of these models. And so, you know, it's kind of the tip of the HPA axis, but we see some evidence for some really interesting differences there. And so we're starting there. And I think, you know, part of that is there is some interesting hormonal response data, but also really elegant work from Jadip Bains and many others shows that those PVN CRH neurons can actually coordinate behavior on a very rapid time scale. And so we're thinking about all of those sort of aspects as we think about it. I think, you know, I know Kathy Grant's group at OHSU has delved more into the idea of HPA axis and involvement in alcohol use disorders in their macaque, their rhesus model. And we've done a lot of recordings with them. And so we see a lot of what I would call nearly identical changes in function in the BNST, but that doesn't really speak to the potential role of HPA or an autonomic system in that response. So I think it's a good area. I think mice are a tough sell for doing some of that work. I think it's better in rats. You just have more space to work with, for lack of a better term.
Right.
And I think, you know, the actually doing it in really dense longitudinal primate studies, looking at these responses in autonomic systems is absolutely the way to go for really getting at this. So I'm excited that Kathy is doing some things like that as well.
Perfect. Thank you.
Sure.
Shifting gears a little bit,
Yeah.
we also reviewed some of your work in related to anxiety and BLA, BNST pathways in anxiety, particularly your paper in 2016 by Nicole Crowley. So the amygdala and the BNST have been conceptualized previously in some work as mediating proximal predictable threats versus distal or unpredictable threats, especially with some fMRI findings in monkeys and humans, providing some evidence for this framework. So how do you see your findings implicating, for example, activation of a BLA, BNST pathway and reducing growing anxiety within this broader context?
Yeah, I think it's a good question. I don't have a great answer how it relates to the relationship. I would point out, I think work that might speak to it a bit better, which is not, doesn't speak to the amygdala BNST coupling, is there's work from a collaborator, Andrew Holmes, as well as Steve Marin's group arguing the BNST, and I believe they both show PFC BNST interactions as playing an important role for response to, and I don't know if I'm supposed to call it ambiguous threats or partially reinforced threats or something. But basically, if using a fear paradigm, if you only have a shock half the time, is what the Holmes lab does, it appears that PFC BNST appears to constrain responding. And so I think in our models that might be playing a role. I think also that we haven't really explored deeply, but part of the amygdala BNST coupling could also be central amygdala coupling between these structures, and we know that they're connected, right?
Right.
We've done that. Other people have done it. Bob Messing's group actually had a really elegant paper in the CRF Cree rats parsing out some of the neurochemical signaling responsible for those interactions. So if I was a bet man, I would wager that the CEA BNST coupling is probably playing is driving that response in human imaging studies versus the BLA BNST.
Gotcha.
It's easy because I don't have to, I don't do the work that really speaks to that. But if I had to guess, I would say that would be probably driving it. And we've started doing, we did some work there early, and we're reinvigorating some work there, looking at dynamics of responses and how information's routed at various points. So I think that's where my money's at for that particular company. I think Ned's group actually has data that speaks to that more directly in the non-human primate model, unless I'm wrong. But I think that's probably likely.
Perfect. Thank you. Very insightful. Thank you again, Dr. Kash.
Sure. You're welcome.
Thank you, Nagal, for those good questions. We have some questions from those who are tuning in, but I wanted to ask one question first, Tom. In some of your data, there were some, it looked like quite pronounced individual differences, even within these small samples. And so, for example, in one set of experiments where you were looking at the effect of reductions in habenula activation in response to drinking, it like there are some animals that didn't even show a reduction at all, and I wonder if you've begun to look at any of those individual differences and what their correlates might be.
Yeah, so that's a really great point. I think that's something that I'll say we're appreciating more and more, is that these dynamic in vivo studies give us the ability to track individuals over time. And so, in the habenula specifically, there was there's one of the, it's 7 or 6 animals. One showed very little modulation of the signal, and then, that is the lowest drinking animal. And so we've started to explore some of these correlations, and, for that, there appears to be a correlation with drinking. I think for all of these responses, there's probably a real wealth of information we have to get at. And I think what I didn't show is we actually have a lot more in vivo, or Meg has done a lot more in vivo recording data, and she's using machine learning algorithms to parse out different behavioral events and align various align the dynamic feature recording features with individual behavioral kind of events. And so we're hoping that, really densely looking through this data and applying these relatively unbiased paradigms and aligning with activity, we'll be able to pull out more meaning from these individual differences. And, part of it also is that, some of the habenula is relatively, lateral habenula, it's small, but it's relatively homogeneous. So we probably don't have as much kind of site-specific effect, whereas the BNST, it's bigger and there's more landscape, and I feel like there's more substructures we might be hitting. Even if we're off by a little bit, we could be hitting, for example, like the fusiform nucleus, which is different than the ventrolateral nucleus. So I think there's a lot we have to learn here, and I think that's the daunting and exciting part of this work, is that we're able to really dig into this more than I think, from my naive perspective, a lot more than I thought we would able to, but I think that's also exciting.
Yeah, if I could ask a quick question too.
Sure.
Tom, the sex differences are really interesting. And have you had a chance, or have you thought about sort of the developmental course of the sex differences, prepubertal through adolescence, and whether or not these are differences that are detectable prior to puberty?
That is a good question. I've not thought about that. I think for some of the manipulation work, it'd be hard to do to get viruses and fibers. But for the generic for the behavioral work, we could look at that, and I think it would be really interesting to parse out when do these differences actually emerge, because it's really stunning to see some of these differences. Like reliably seeing a difference is really exciting. If we could say, okay, this is when the paths diverge, or do they diverge early? Is it an organizational sort of difference, or is it a hormonal difference? I think that's a really good question. I had not considered. I'm sure Meg has, but I had not considered. But I feel like it would be good, because also we know that the risk profile for alcohol abuse is not for it's great during adolescence. So it'd be good to know something about these sort of sex differences in alcohol use, the sequelae of behavior we see at that time point. So I feel like, that's a really good idea.
So, Tom, there are a few questions from the audience that I can read. One is from Layla, who said, "Beautiful talk. Previous literature indicates that noradrenergic inputs to the BNST are involved in stress-induced relapse and other addiction-related outcomes. How do you think that the noradrenergic and 5-HT systems may converge?"
So that is a great question. My collaborator here at UNC, Zoe McGilliot, so she has previously focused on norepinephrine and BNST, and so I have, I don't work on norepinephrine basically. She's got that covered. I know she has been talking about launching a new investigation, and we've been talking about it as part of a collaborative grant we're part of for an alcohol stress consortium, looking specifically at noradrenergic signaling in the BNST and some of these similar behaviors. We haven't done really a lot looking at the interaction, partially because it's just now that we're getting the tools that give us capacity to look at the dynamics, I would say. So we have the Grab 5-HT sensor. There's also been a Grab NE sensor. So I think we're finally at the point where we can drop in sensors and look at the dynamics of responses. I think it would be really interesting to kind of combine this to use, we know we can optogenetically shoot out, we can stimulate serotonin release is what I meant to say. We could also, I know Zoe's group has looked at, Dr. Mabelot's group has looked at optically evoked norepinephrine as well. So it'd be really exciting to kind of look at how they interact. We haven't done it, but I think there's probably a lot to it, because there is a wealth of literature about BNST, norepinephrine. There's really interesting, I would say, clinical data on the alpha-1 adrenergic antagonist, prazosin, how that can impact alcohol use disorder phenotype. So I think there's a lot of possibility for that. You know, as you add things, it gets a little bit more complicated, but that's the nature of biology, I would say. Great question, though.
Let me see if I can squeak in one really short question
Sure.
from another person in the audience, Ankush. He asks, did you check if the estrous cycle mediates the sex-induced changes in the mouse behavior?
That's a great question. We have not looked at that yet. And so, you know, I think that it is on the docket for something useful to look at. Because we're seeing a robust sex difference between the groups, it wasn't our primary endpoint. I think it would be, or wasn't part of the primary design. I think it would be of value to look at that. I would note we had another recent paper looking at dopamine in the BNST and pain, and we spent a lot of time looking at estrus and never saw a relationship. There was a robust sex difference that had nothing to do with it. Nothing measurable to do with estrus. So I think it's a good question, and we'll do it. But also, it wasn't the first thing we thought of.
Yeah. Okay, I think our time is up. I want to, for all of us, thank you for a great talk. Really appreciate it, and look forward to having you join us later this afternoon.
That's great. Thanks again, Tom. And we've got a 15-minute break, and then David Lewis is going to take up the mantle next. It's going to be an exciting talk related to schizophrenia and its pathophysiology. So let's take a bit of a break, and we'll be back.
Fantastic. Thank you. Bye-bye.
Thanks again, Tom.